we're gonna get started here. We've got Jordan here in person, and we'll see if anybody else comes in person. But really, this is a lab in the sense that you are going to be using data that you collect about yourself to figure out if you're a taster, right? It's not a super complicated lab, although you do need this PTC paper to do it, right? And you can't get this on your own. You can only get this if you're a school. So that's why I asked people to come in, and we did have about 10 people come in and get the paper. So basically, we're going to be reviewing just a little bit about genetics here. Due to the holidays and the way that it kind of worked out, I didn't get to do as many virtual sessions on genetics as I'd like. I hope that you guys saw the video from last year that I sent out that's super long, but is a really good review of genetics. If you did not see that, you can go to modules in the biology class. Maybe you can go to modules. And at the very bottom in the virtual session and labs section, oh, my computer is not working right now. There we go. You will see that I did publish a genetics review virtual session from last year. Right, this is totally optional to watch, but I know genetics is a pretty hard and interesting topic, right? So it's not like you're going to have to do these exit tickets or anything like that. But there's a PowerPoint here. I go through a lot of the vocab and the crosses and how to do those things, how you get genetic diversity, etc. And some of these are questions on the quizzes and tests. So you could always check this out before you take the test. If you already took the test, you might want to check it out before you take the final exam. Also, there's 2 PowerPoints that students last year made about Huntington's disease and sickle cell anemia, which are 2 pretty interesting genetic diseases that you might want to learn about, sort of what those are and how they work. So that's a good review from last year. And it's an hour and 20 minutes long, but 20 minutes of it is people making those PowerPoints and me sitting there staring at the computer. So you don't have to watch that part. You can fast forward through that part. So what we're going to do today is we're going to talk about your genetics specifically with the PTC paper. And I know some people have already tasted the paper when they came in. The rest of us will taste it in a moment. So we're going to review a few pieces of genetics vocabulary. We're going to observe a trait in ourselves using the PTC paper and then predict the possible genotypes of your parents. And then we're going to complete the DBA for genetics and heredity unit. So let's start with a warm-up question here. This was something I saw a lot of people getting confused, these questions, which is why we're going to go over them. So the first question is, what is the difference between protein synthesis and DNA replication? And the second question is, are all mutations bad? Explain. So take a second right now, think about those 2 questions. And propose some answers in the chat here. And there's the questions there, Jordan, if you need to look at them again. But what is the difference between protein synthesis and DNA replication, and are all mutations bad? Okay, I see Alina wrote yes. I'm not really sure what question that's answering, since one of them says explain and one of them says what is the difference. Elizabeth says protein synthesis is like the making of proteins, and DNA replication is when DNA is copied. Good, right? So DNA replication and protein synthesis are 2 separate processes, right? And it's a little confusing because the lesson that they have for this in Canvas clumped them together. Right, your assessment was to talk about protein synthesis, but the assignment was called DNA replication. And I know that's a little confusing. The reason that they're grouped together is because DNA replication is really similar to that first step of protein synthesis, transcription. right, where the DNA is getting copied by the RNA. That's very similar to DNA copying itself, right? It's not exactly the same, but it is a similar process. So keep in mind that those are two separate things. DNA replicating is going to happen during mitosis, right, during meiosis, and if your cell is repairing DNA, sometimes it might have to replicate parts of it. Otherwise, that's it. Protein synthesis is happening all the time. You're making proteins right now. So protein synthesis is an ongoing, forever process. You always have to keep making more proteins, replacing proteins, etc. And DNA replication is not happening as often, I would say. All right, then what about our second question? Are all mutations bad? Jordan's shaking his head saying no. I would agree with Jordan. Sure, Ariella, go ahead. So Jordan says no. I'm sure a few other people think no, but why? Why are they not all bad? The X-Men mutations are not bad. Okay, well, possibly. We could maybe all agree with that. Yeah, Ariella, if you're talking, I can't hear you. Destiny says some mutations can be beneficial. 100% correct, right? Mutation sounds like a bad word, right? Of course it does. You think mutation, ooh, I don't want that, right? It really just means a change in the DNA. There's three types of mutations. There's mutations that literally do nothing, right? Sometimes the DNA gets changed, no result. The protein's not changed, nothing matters. In fact, you have a ton of those mutations, right? We call them variations usually, in your cells, right? Your DNA is not going to be 100% matched to another human unless you're a twin. And then even then, it's still not usually 100%. But there's also mutations that are good, right? A mutation that might cause you to be better. And that's what we're going to talk about in the beginning of the second semester when we talk about evolution, right? If we don't have these variations in the population, then species cannot evolve. So those mutations can actually cause suddenly maybe someone to be faster, stronger, smarter, etc. Right? That's kind of looping it into a pretty vague context, but all these little mutations can add up and actually end up benefiting an organism, right? Or they could be the complete opposite, where a mutation is completely detrimental, right? It could kill the organism, or it could be something that causes disease. So have you guys ever heard of lactose intolerant? Some of you might be that. Here's how you spell it. I'm putting it in the chat just because sometimes when we hear it, we don't know exactly what that word looks like, right? But lactose is a protein. Those proteins are found in milk, right? In cheese, some types of cheese. If you're lactose intolerant, you actually don't have an enzyme or a protein that can break down lactose. So what happens? You can't break it down, it makes you sick, and then you usually have diarrhea, right? That's a mutation. Not being able to create that lactose enzyme is a mutation, right? Is it the worst thing in the world? No, right? You could totally survive and have a normal life, and you could even still eat cheese and milk, right? You're just gonna feel sick after. Other mutations, like one of the PowerPoints, which I hope you guys go look at with Huntington's disease, is scary, right? That's a dominant gene. You don't know you have it until you're in your 40s, when suddenly it gets activated and basically your brain and nervous system completely deteriorates and you lose all your ability to think and your memories and function, and eventually you die. Right? So there are other mutations that result in awful things, and not even like a quick death here. We're talking about like suffering for years. Whereas lactose intolerance is annoying, but not the worst thing ever. What about lethargy? So being lethargic is not necessarily genetic, right? It could be caused from a lot of different things. And that's where genetics becomes hard, right? Because it's hard to figure out exactly what is causing something. Especially if there could be like 10 factors causing something, it's almost impossible for us to figure out which one. All right, so another reminder here, make sure you're looking at the feedback that you get on your assignments, right? Sometimes your feedback might just be how great you did or one thing that you need to fix, but you can always go back and redo those assignments. So if you did your DNA replication assignment where you actually wrote about DNA replication, I probably gave you some feedback to change it to protein synthesis, right? You can always resubmit that assignment and get more points for that. So be thinking about that, especially since we're going to finish this course pretty much by December 15th. January is going to be for revising and also for working on your project, your collaboration project. So you can resubmit any assignment. Quizzes and tests, you cannot resubmit without my permission, right? So if you're looking at a test and it's looking really sad and you need some help with it, then you can email me. And Charlie says, I want to redo a bunch, but I don't see a way. So Charlie, send me an email and I can send you a video about how to resubmit. If you don't have a way to resubmit, you can always email me, right? I know Canvas made changes where they actually possibly made it so that once you put a grade in, you can't submit stuff anymore. But I've had people resubmit, so I think you can. But just keep that in mind, right? I'm giving you that feedback so that you have the opportunity to correct what you've done. You don't have to just sit there and look at it and say, oh, well, I got that wrong. All right, so genetics. Are you a taster? We're going to get into the PTC fun. So most of these things are things you should have already gotten out of the lessons and could possibly be a review here. But we know that some traits are inherited in a dominant and recessive manner, right? If you get the dominant trait, it's expressed. If you don't, it's not expressed. We know that we get one trait from each parent, right? So we call those alleles. You get an allele from your mom, you get an allele from your dad. That means you can have a bunch of different combinations, right? You could have a homozygous dominant combination, which is where you have two dominant traits. Homozygous means the same allele. You could have a heterozygous combination where you have a big T and a little t. I'm using T as an example. It could be any letter here, where you have one dominant trait and one recessive trait or allele. Or you could be homozygous recessive, where you have two recessive alleles, right? Those are the combination possibilities. If a trait is truly a dominant and recessive trait, right, then you only have two possibilities. You either have the trait or you don't. Now, we know from our studying of genetics, you might even know from going through some of the lessons, that not every trait works like this, right? That would be amazing if it did. It would be so easy for us to figure these things out. Sometimes you can have co-dominant traits where it's kind of expressed, which we'll talk about that as we talk about PTC as well. You could also have traits that are controlled by multiple genes. So for example, eye color, controlled by many genes. Height, controlled by many genes. So there's no way to easily predict. That's why if you have tall parents, you might not be tall, because that trait of being tall is not expressed by just one gene. It's many, many genes working together. So that's when it can become super blurry. Right now, there are some dominant and recessive traits that are super easy to see, right? Widow's peak, when your hairline actually peaks down. I don't have a widow's peak, so my hairline is straight here, right? If you have that peak, that's a dominant trait. My earlobes are attached to my head, right? There's no like little space in between. That is a recessive trait. If your earlobes are detached, that's a dominant trait. Everybody take your hands, cross them on top of each other. Look at what thumb is on top. If your right thumb is on top, you're recessive. I'm recessive. If your left thumb is on top, that's a dominant trait. Right now, all those things are like, seem kind of silly. Does it really matter if your earlobes are attached or if your right thumb is on top? No. Right? But those are things that we have identified as being inherited directly through a dominant recessive manner. That means if you go to your parents and you ask them to cross their hands, right, they're going to have something that shows you that you're related, essentially. They'll have traits that you could have inherited, if that makes sense. Yeah, so if you have blue eyes and your parents have brown eyes, totally possible because they, like I said, multiple genes control eye color. So you could have inherited recessive alleles in many places and then ended up having blue eyes, right? So eye color is especially a big misconception because most people think it's a dominant recessive thing and it's not. So it's really hard to follow and trace that stuff, right? Because blue eyes could not be in a family for 5 or 6 generations and then suddenly appear. And that's totally possible, right? That doesn't mean that you're like not in the family. So keep that in mind, right? And there's tons of information about this. So any traits that you have that you're interested in learning about, you can always research and you can read. And now that you know this basic vocabulary, right, you will be able to interpret what those things mean. So if you find out something is controlled by 7 genes, okay, that's going to be pretty hard to figure out exactly which genes you have. So for our PTC paper, PTC is this chemical called, let's see if I can even attempt to pronounce it, guys. It's pretty hard. Phenylthiourea phenylthiocarbamide, which we call PTC, which is much easier to say. It is a harmless chemical, right? But it basically mimics a poisonous plant chemical. It's not poisonous, don't worry. I want to give you a poisonous thing to taste. They wouldn't sell me a whole vial of poison that easily. So basically, we'll talk about kind of how it was discovered and all that in a moment, but it's just a piece of paper, right? And if you can taste it, you will know because it tastes really disgusting. And if you can't taste it, like me, it just tastes like paper, right? So when I put it in my mouth, it literally, I feel like I'm eating paper. When other people put it in their mouth, depending, right, on how sensitive they are, which we'll talk about, they literally, like, I did it to Miss Dowell. I made her taste the paper, and she was like, oh, and had to drink a cup of water, and she said she could taste it for the rest of the day, right? So be advised, probably have some water nearby if you haven't tasted your paper yet. I know a few people came in and did taste the paper already and found out if they could taste it or not, right? So if you haven't, and you did pick up a piece of paper, go ahead and pop that in your mouth right now. You don't have to eat it, right? Literally, you could taste it and take it out. It doesn't have to be a long-term thing here.
Good sense that there's something on the paper, but it doesn't have like any severe taste. It tastes like tasteless flour.
Okay. So Jordan's a non-taster. It just tastes like paper. What about you, Victoria? What was your experience? Could you taste it? Elizabeth? I think Charlie came and grabbed the paper. Charlie could taste it, I remember. Victoria could taste it. Okay. Elizabeth couldn't taste it. Okay. Right. So what we would expect to see in our population, Alexandra is a non-taster. Great. It tastes very bad. Yeah. Right. So people describe it as tasting awful, like really bad. You would not eat it, which is crazy because I would totally not even notice. Right. It literally tastes like paper to me. If somebody poured this chemical all over my food, I would just be eating away and not even notice. So we'll talk about why that's important in a minute. But basically, if you are a taster, right, if you can really taste it, it was awful. You most likely have 2 dominant traits. Most likely, right? Of course, taste is perceptive, so that can make this complicated. If you could taste it, but it was gross, but like it wasn't the worst thing in your life, you're probably heterozygous. You probably have 1 dominant trait, 1 recessive trait. If you couldn't taste it, then you are, like me, homozygous recessive and a non-taster, right? Which means that we have 2 recessive alleles. We can only pass on recessive alleles. So, pretty cool. You now know about your trait. Now I use T arbitrarily because for taster, right, to represent taster. We could have used any different combination of symbols here as long as the capital was representing dominant and the lowercase was representing recessive. So if we were to do a Punnett square for this, because I know some of us have done Punnett squares before, right, where we would actually make our boxes. I know a lot of us did not do the dihybrid crosses correctly, but that's a different video situation here. If we do a cross here between a big T and a little t and a big T and a little t, there we go, we will end up getting the ratio that we would expect to see in our population. It's pretty hard to draw with the mouse here, guys. This is a hard thing for me. Notice how weird my T's look. So if we do this type of cross, it will help us predict what we would see in the population, and it's pretty right on. We would see about 75% of the population is able to taste, right, with 25% being dominant, homozygous dominant with 2 dominant traits. And then we would see 25% of the population being a non-taster. So if you're a non-taster, you're part of that 25%. We don't see that exactly perfectly. It's going to depend, of course, on which part of the population you're sampling, right? In our class, since we only have like 5 or 6 people who tasted it, obviously our numbers are too small to really get a good concept of that. But know that 75% of the population can taste this.
So that would be like a genotype then.
Mm-hmm.
So if I wasn't a taster, I would write homozygous recessive.
So genotype, good question. So what is the difference between genotype and phenotype, right? Genotype literally means the letters. So if you're a non-taster, your genotype is little t, little t. Phenotype, you could associate the P with the word physical because, I should have just done a text box and typed this, but physical, because that's actually what you see. So your phenotype is going to be whether you're a taster or a non-taster. Your genotype is going to be the letters that represent your genes. Now keep in mind, these letters are representing the genes. If we looked at your genes, we're not going to see little t's or big T's on them, right? It's a representation of the gene. So the genotype is going to be the letters, and the phenotype is going to be the trait, whatever it is. So in this case, if you're a non-taster, your phenotype is that you're a non-taster. Your genotype would be little t, little t. So let's go into, so Elizabeth, we're going to talk about parents in a second. That is a good question. That's actually going to be our exit ticket here. But we're going to read a little bit here. We're going to do a little article reading about kind of how we found this out, and also what it might mean in terms of evolution and why people can taste this chemical in the first place. Also a good reason to do a text box because erasing these is pretty hard too. There we go. Okay. So what is PTC? Why can we taste it? What is even mattering here? We're going to read this pretty short article together. So PTC, the genetics of bitter taste. I'm going to read the first paragraph to you guys, and then we might actually take volunteers to read, so be thinking if you would like to volunteer. In 1931, a chemist named Arthur Fox was pouring some powdered PTC into a bottle. When some of the powder accidentally blew into the air, a colleague standing nearby complained that the dust tasted bitter. Fox tasted nothing at all. Curious how they could be tasting the chemical differently, they tasted it again. Now, let's pause and note that this is definitely not advisable to be tasting chemicals in a lab, but in 1931, people were doing that. Thankfully, because then they figured out what stuff we shouldn't be tasting. The results were the same. Fox had his friends and family try the chemical, then describe how it tasted. Some people tasted nothing, some found it intensely bitter, and still others thought it tasted only slightly bitter. Right, so we see a picture here of what the PTC paper looks like now. And we see some pictures of what bitter things taste like, right? Coffee is super bitter. Dark chocolate with no sugar in it is very bitter. A lot of vegetables are bitter. I love bitter stuff. Now I see why, because I can't really taste bitter stuff, so it tastes good to me. So the PTC gene. Would anyone else like to read a paragraph, or do you guys want me to keep reading? Elizabeth can read. All right. Do you want to start with the soon after its discovery one?
Yeah, up till it says 2003.
Yep.
Okay. Soon after its discovery, geneticists determined that there is an inherited component that influences how we taste PTC. Today, we know that the ability to taste PTC, or not, is conveyed by a single gene that codes for a taste receptor on the tongue. The PTC gene, TAS2R38, was discovered in 2003.
Great. Anyone else want to read? All right. I'll read again. I don't mind. I kind of like reading out loud. It's fun. There are 2 common forms or alleles of the PTC gene and at least 5 rare forms. One of the most common forms is a tasting allele, and the other is a non-tasting allele. Each allele codes for a bitter taste receptor protein with a slightly different shape. The shape of the receptor protein determines how strongly it can bind to PTC. Since all people have 2 copies of every gene, combinations of the bitter taste gene variants determine whether someone finds PTC intensely bitter, somewhat bitter, or without taste at all. So what they're saying here is that there's a difference between our homozygous dominant and our heterozygous tasters, right? Like I said, if you're homozygous dominant and you have 2 dominant copies, you super taste it. It is awful. Otherwise, if you only have 1 copy of the dominant gene, you might taste it, but it's not as intense. Now, of course, this is hard, right? Because again, like I said, taste is subjective. You might think something tastes really, really bitter, and somebody else might say, doesn't taste that bitter to me. So that can make this hard to figure out as well. Now we'll look at some of the science behind this, right? You got your tongue, all the little taste buds. So basically what's happening is you have a cell receptor, and our cell receptors, this is kind of how we draw them in biology to represent them. They look like little football goals, and they're on the cell. So here's our cell, and there's our receptor. Now, if you can taste PTC, that means the receptor is shaped so that when the PTC comes in, the PTC is the same shape as the receptor, right? Oops, I didn't mean to make that crazy line there. So the PTC can fit into the receptor gets activated, and that tells your cells the taste, right? Which then goes through your nerves to the brain and all the rest. If your receptors are from the recessive trait, right, you're a non-taster, your receptors are a different shape. So they might be more like this shape, where maybe the PTC doesn't really fit in there, right? It's too small possibly, or too big, right? It could be a bunch of different kinds of shapes. Obviously, we're talking about 3D things here, so it's much more complex than what I'm drawing. But this is the concept behind it, is that the receptor is either activated or it's not. And that's how you're able to taste. And that's actually how a lot of stuff works, is through cell receptors, right? Even insulin, type 2 diabetes, it happens because the receptor is not recognizing insulin anymore. Okay, so just a little bit more here. So natural selection at work. This is something that we're going to talk about next semester when we learn about evolution. But although PTC is not found in nature, the ability to taste it correlates strongly with the ability to taste other bitter substances that do occur naturally, many of which are toxins. Plants produce a variety of toxic compounds in order to protect themselves from being eaten. The ability to discern bitter tastes evolved as a mechanism to prevent early humans from eating poisonous plants. Humans have about 30 genes that code for bitter taste receptors. Each receptor can interact with several compounds, allowing people to taste a wide variety of bitter substances. So, back in the day when humans were scavenging, walking around, literally picking up plants off the ground, eating them, right? People who were able to taste that bitterness, imagine if you put something in your mouth that tasted like PTC, for those of you that could taste it. If you put it in your mouth, you were like, oh, not eating it, right? Meanwhile, me, I would have been like, cool, this plant tastes great. And then I probably would have died, right? So that's kind of how they figured out not to eat bitter things, was like, oh, people that were eating bitter things were dying. That means poisonous, don't eat it, right? Today, it really isn't mattering so much, right? Because we obviously are not buying poisonous plants at the store. And most of us are not going out into nature and collecting plants to eat, right? So we do see that recessive trait still existing. Back in our ancient population of humans, if we would have sampled the population, we would have seen a lot less people with that recessive trait because those people would have probably been dying, right? Not necessarily. Not everybody obviously died, but some people for sure did. So you guys can even think about this with vegetables. So a lot of people hate like kale, right? Brussels sprouts, broccoli, because they're bitter, right? And people don't know necessarily that they hate them because they're bitter, but they're like, ew, I don't like how it tastes. They are bitter, right? And they are kind of reminiscent of poisonous plants. So some people who are really sensitive to PTC actually have a really hard time eating vegetables. Fun fact. So here is what the structure of PTC actually looks like, right? In case you're interested in that kind of stuff. It, like I said, it resembles a toxic substance that's found in plants that are poisonous. So if the ability to taste bitter compounds conveys a selective advantage, i.e. you're going to survive because you're not eating poisonous stuff, then shouldn't non-tasters have died off long ago? Why do so many people still carry the non-tasting PTC variant? Some scientists believe that non-tasters of PTC can taste another bitter compound. This scenario would give the greatest selective advantage to heterozygotes, or people who carry one tasting allele and one non-tasting allele. Right? So this is what geneticists do, is they try to figure out how have these genes changed over time based on the population of people. So they're saying here that having one dominant and one recessive could possibly have been the most favorable in any environment, because maybe you also had the ability to taste other bitter things, right? I can still taste bitter stuff even though I can't taste PTC. It's not like I can't taste bitter things at all. And then, let's see. Oh, what a cute little monkey. PTC's sensitivity is often used as an example of a simple Mendelian trait with dominant inheritance. However, tasters vary greatly in their sensitivity. And while the PTC gene has about 85% of the total influence over whether someone is a taster or a non-taster, there are many other things that affect PTC tasting ability. Having a dry mouth, right? Your sensitivity can change over time. So, for example, as you grow up, you try different foods, right? You become accustomed to taste. For example, you might like the taste of coffee suddenly. Maybe when you tried it the first time, you didn't. Right, you're acquiring a taste for something, and that actually changes the structure of your taste buds. So that could influence whether people could taste PTC. So essentially, there's a lot of stuff going on here. Yeah, exactly, Destiny. I used to not like dark chocolate. Now I like it. You've acquired that taste for that bitterness, right? So that could influence your concept of how bad the PTC tastes. All right, and then potential health applications. There's also, this is an interesting connection too, that studies indicate that individuals with strong taste, that are strong tasters of PTC gene variant, were less likely to be smokers. This may indicate that people who find PTC bitter are more likely than non-tasters to find the taste of cigarettes bitter and may be less likely to smoke, right? Another possible correlation. Interesting thought. Meanwhile, seems crazy to me because that's 75% of the population that can supposedly taste PTC, and there's a lot of people that smoke. So, you know, that's kind of one of those things where you're like, well, either it doesn't matter that much to them, or maybe only a few people are impacted by that. Right, but this is the kind of stuff that geneticists do, right, is sort of figure out these connections between lifestyle choices and genes, essentially, right? Because environment does have a huge impact on genes. The fact that you can acquire a taste, right, should tell you that even though your genetics may say one thing, your environment can influence those genes to change, literally change structure, which is pretty interesting. Okay, so that was a little background here about the PTC. So, okay. Let me get this back in full screen mode here. So then the last part of this here, before we do our DBA, is this is the exit ticket, right? Which is to figure out your genetics. What is your genotype? What is your phenotype? right? So again, what are the letters? What are you, a taster or a non-taster? And then what are the possible genotypes of your parents? Possible. Now, the only way you could for sure predict, and I'm still saying predict because we would want to confirm with an actual genetics test, right, what your parents could do, you would actually have to give them PTC paper, right, and have them taste it as well. So what you want to do is think, okay, if I am a little t, right, that's me, that means I had to get a little t from each of my parents. So my parents must have, each of them must have at least one little t, right? They could both be little t, or they could be big T, little t, both of them, right? Or one of them could be big T, little t, and the other one, little t, or vice versa, right? So there's a lot of different possibilities of this. So I want you to try to list out all the possibilities, and I'm going to erase that so that people cannot copy that from me exactly, right? But that's the concept here, thinking about where those alleles came from and what possibilities your parents could be. Neither of my parents could be big T, because I don't have any dominant traits. Where would that dominant trait have gone, right? So Alina thinks maybe you're big T, that's fine, right? If you want to take a guess about your genetics because you didn't actually taste the paper, but you think, oh, bitter stuff I really hate, maybe I am a PTC taster, that's fine, right? This is really more of an exercise in trying to predict what genetics your parents could have based on something you have. If you don't want to predict what your own thing is, you can use my results, right? I'm a non-taster. So that's going to be the exit ticket that you guys fill out in Canvas, right? It's in there, it's available for you to see. I will be uploading the PowerPoint as well as this recording. So the DBA, we are all going to do, I've done this in my other classes and I haven't done it in biology, and I realized how silly that was. So let's see, this is the PTC article, which I've opened multiple times. This is, there we go. Okay, so I'm gonna post the Google Doc link in the chat, right? I'm also gonna post the code word in the chat. It's a hard one, so you might wanna write that one down. So in the chat right now, you should see, and I'll tell you the code word, Jordan, so don't worry. You should see a link to the Google Doc, and you should also see the code word. So when you get into the Google Doc, if you want to go ahead and add your name to a box, we're going to be writing answers to the questions. I also already put the questions in there. So if you would like to start answering, you can. I am going to read them out loud, and I will also give you guys some background on some of these things. Yeah. So you are just going to tell me your answers. Okay? Uh-oh, we got two people in the same box.
Do you have to do this from memory, or can we use it?
No, you can use your notes. And you can listen to the questions, and then if you want to jot down answers and then share them with me, that's probably going to be the easiest. Okay, so the first question is, what are two differences between mitosis and meiosis? You can use bullet points here. You could write in full sentences, right? It's up to you, but what are two differences between mitosis and meiosis? Great, I see a lot of people saying 2 rounds of division. The number of chromosomes is reduced by half in meiosis. Great.
Mitosis is asexual and meiosis is sexual reproduction.
Excellent suggestion by Jordan over here. Mitosis is asexual, meiosis is sexual. That's two differences, I guess we could say, right? Yeah.
Yeah, the asexual contains 2 daughter cells, and then it's 4 daughter cells.
Good. Makes 2 cells versus 4 cells. Do we have enough boxes here? Because we could put some more in here if you guys need more boxes. I'm going to just put your name on here, Jordan, so that I remember to add in a grade for you as well. Nothing is popping up on your screen, Victoria. It could be because we have like 1 million people in this document. It might be lagging. So Victoria, do you want to just write your answer in the chat and I'll actually post it into the document for you? Okay, so the first question is 2 differences between mitosis and meiosis. All right, meiosis has 2 rounds of genetic separation and cellular respiration. Okay, I was gonna say, and cellular respiration. Mitosis has 1, mitosis has 2 divisions. Excuse me, meiosis has 2 divisions. 2 daughter cells are reproduced. Okay, it's looking good. So yeah, guys, keep in mind the 2 main differences are that mitosis makes a direct copy. And you know how I remember that one? I think, my toe! Like when you stub your toe and your toe's bleeding everywhere, you have to repair it using mitosis. And then meiosis, it looks like the word me, right? And how were you created? By 2 different parents, right? So that's how I remember the difference between the 2, if that helps you. I know it's a little ridiculous, but sometimes it's hard to keep them separate, right? They sound very similar. Okay, awesome job, Victoria. I'm gonna add that in for you. And of course, you also wanna be thinking about crossing over, right? Crossing over, or this concept that the DNA actually switches places in order to create more genetic diversity, right? That's super weird and important. And it literally does, like pieces of the DNA break off and rearrange. Okay, so some people still working on the first question. That's great. I see some people already starting on the second one here. The second question is, who was Mendel? Which I apologize, I should capitalize his name because he is a proper noun. And why is he so important? Yeah.
He was the guy who really figured out, oh snap, this is how everything works together. This is how we become who we are. So.
Yeah, exactly. He was the father of genetics, right? He was a monk, I see a few people wrote. Yeah, and he wasn't a scientist, guys, and he figured this out. Alina, you can type your answers in the chat as well if you can't get them to type into the Google Doc. Yeah, and I'm gonna leave it open for a little bit after so you can always finish later if you need to. Right, he figured this stuff out. He grew pea plants and he basically did selective breeding. He bred them and figured out how these traits were passed down. He was the first person to come up with this idea of dominant traits and recessive traits. And in pea plants, there were tons that were so easy to see. Yellow seeds versus green seeds, round seeds versus wrinkled seeds, right? Different heights, etc. And they were all expressed dominantly and recessively. And he actually wrote all that out. He literally had like pages and pages of him documenting all of this information about plants. So pretty interesting guy in the sense that he literally, all of his data actually led to us figuring out that there was such a thing as genetics. And the crazy thing is, guys, he didn't even know about genes. He didn't know about DNA. That stuff didn't even exist yet. DNA wasn't discovered until the 1950s. Right? So he came up with this idea that somehow these things were being passed down. They didn't really know how. Right? And that's why genetics really didn't become a super big deal until pretty recently, because now we know enough that we can actually start understanding and applying these tools. So the last question is, what are some cons at least 2, of inserting genes into organisms to genetically modify them? Ooh, I like Destiny's thought here. All organisms will have the same genetic makeup, and if a bacteria would attack them, then they're all going to be affected, right? So that's one reason, is we're eliminating natural variation, right? That's a problem. Any other thoughts? By adding genes, we do not know which genes we are altering. Excellent. Right, so guys, do you know how they insert genes? I know you're typing and thinking right now. They use something called a gene gun. Literally a gene gun. They take a gun, they shoot the gene at the DNA of the organism and hope that it gets accepted. Guess what? They disrupt other genes when they're doing that. They don't know what they're doing, right? The other thing is genes are incredibly complex. They are not like, oh, here it begins, here it ends. There is a beginning and an end of the gene, but then a mile away on the DNA, there could be something that actually relates to that gene. A mile the other direction on the DNA, there could be something that relates to that gene. And I say a mile because I mean like very far. Genes are not super broken up into sections, right? They have multiple, it's called epigenetics, but it's basically they have other parts of the DNA that actually control them that are not necessarily part of the gene.
So what question was this?
The third one.
Which was?
2 cons of inserting genes to genetically modify organisms. So Victoria says another gene could be messed up and disease could come to the gene. Yeah, exactly true. Right, and that's actually usually what happens. And a lot of people say, well, why aren't we doing this to humans? Right, because if, for example, we know somebody doesn't make insulin because they're a diabetic, why can't we just insert insulin genes into them? Well, hopefully eventually we can, right? Because that would be cool. That would be an easy fix. They're trying to use viruses right now to do that, right? Because viruses can actually infect cells and change DNA. So they're trying to use viruses in a positive way. Why can't we use a virus to infect cells with a gene that's actually going to help someone? So that's kind of a new forefront on the science field. But essentially, let's see, sometimes if the modification isn't a harmful mutation could occur and cripple the subject. Yep. We're going to eliminate, yeah. And guys, one of the things that is a problem with this that seems kind of obvious is that scientists are basically playing God in a way here, right? Which is kind of scary. They're altering the natural genetics of something, right? And that could be morally wrong in your eyes. You could also think, no, that sounds cool, keep doing it, right? There's multiple views on this, obviously, and neither of them are necessarily wrong. However, it is something to think about, right? Is it okay for scientists to manipulate DNA? Do you think it is morally okay, right? It's a thought. Ariella says it's illegal. The person would have to be willing to give themselves up. Yeah, of course, right? We can't just go up to a person and start injecting them with random DNA. No, this would have to be something controlled. But if you have a serious life-altering disease and it's the only option for you, you might be saying, sure, inject me with DNA all day. So in some ways, this is morally questionable, right? Is it okay for scientists to even do this? We can do it, that's cool, but should we do it, kind of thing. So sometimes we actually end up having questions like that in science, like, is it okay to do it? I don't know. So what do you guys think? Is it okay for scientists to manipulate genes like this? Just from a moral standpoint, a yes or no? Type it in the chat for me. There's no more questions, I'm just asking. Well, what do you think, Jordan? You think it's okay for them to do it?
Well, if it turns out that was like the cure to cancer, well, do you think that you're going to say, no, let people keep dying from cancer because doing this is morally wrong? So you're going to let all these people die of cancer just because it's kind of morally I mean, a lot of people will disagree with that.
No, I think that's a good
So
point.
then the game completely changes if it's something that big.
Right. If it was something that could cure cancer, Jordan says, then would we care if it was morally wrong? I don't know. Right? Maybe not. Christian says if the subjects are willing for it to happen, it's fine. Colton says, yeah, do it. Ocean says depends. Yeah, I think it depends in some ways, right? Why are you doing it? Intentions mean things here, right? If someone's intending to ruin the world, okay, well, then we have an issue. But if they're trying to cure a disease, other people said no, they shouldn't do it, right? Which is a totally valid thought as well, because it is, like I said, there's a lot of issues with it, and it's scary. All right, so that was the DBA, guys. I'm going to leave the document open for a little bit longer, so if you want to edit your answers, if I see anything in there that looks wrong, right, I'll either send you an email or leave you a comment just telling you, hey, maybe this is a better way to word it, etc. And then I'll just go in later and plug in those grades for you guys. Otherwise, check out that other virtual session on genetics, especially if you're interested in genetics. Genetics is one of my favorite things to talk about, so I talked a lot about it. And also next week, right, is Thanksgiving. The school itself, the physical building is closed, but we do have work due on Monday and Tuesday, so make sure that you're still getting that stuff turned in. And then Wednesday, Thursday, Friday, you have off, right? And you can enjoy, or you can use that time to catch up if you're behind in school. All right, so otherwise, I'm going to end this meeting. If you have any questions, feel free to email me. It says we're supposed to fill out a lab sheet. Yeah, sorry, there is no lab sheet. I'm actually going to delete that assignment, Elizabeth, and just have the exit be the only thing, which I'll do right now. So the only thing that you need to do for this lab is genotype, phenotype, and what your parents could have been, and that's it. Okay, I think last year I did have a lab sheet, but it was basically said the same exact stuff. So I was like, why do I have a sheet when it could be three sentences? All right, so I'm gonna delete that PTC paper thing. What if you didn't get the paper? Then you use my results, or you take a guess at what you think you might be. All right, bye everyone. Have a good Thanksgiving.